Hi, I want to talk about my project. It is gaze recurrence quantification for multiple participants in a collaborative environment. People accomplish tasks using tools available to them and usually work in groups when possible. These tools have variety of forms and most of them are electronic devices nowadays. They are interconnected, they have high resolution displays, and they have relatively large scale computation capabilities. But the question is, do they really help us? And if so, how much is the contribution? What if some are distracting or have negative effects in task performance, or some may have overlapping functionalities? Therefore, we need quantification methods. My project is in three folds: setting up a system that will help us collect data and find effective ways to analyze such data, and ultimately offer preliminary results for future refinement. Let's start with the data collection part. Since the audience is limited, I will leave this part here brief. We use Pupil Labs eye tracker with multiple HD cameras. Markers are emplaced on target environments in advance, thus we can distinguish surfaces and calculate gaze points. We use network-enabled computers and they all have synchronized. And post-processing is also available and we widely use this feature. It is possible to collect data in various ways. However, sensing everything available, then processing is not the best approach. We prefer to decide areas of interest in advance, design experiment, and collect data accordingly. Let's consider a scenario with one participant. Tracking the gaze point is straightforward. An eye tracker and markers on a surface will reveal enough data to quantify the attention point. It doesn't necessarily need to be vertical or horizontal setting. And whether the participant is stationary or mobile, it doesn't matter either. It is still possible to track the point of interest by time. Now consider another case where two participants involved and do exactly the same task. They may perform the task consequently or at the same time. When we add multiple participants, the case and also the experiment design gets more sophisticated. So let's start with from very simple experiment and get preliminary results. Let's consider a simple task. Three participants are asked to look at vertical screen and follow the given directions. It's very straightforward. They should gaze at areas A, B, C by following the sequence provided. Each run took less than 30 seconds. We ran single participants at a time, then synchronized runs. Here's the initial plot of two participant gaze points. The figure provides eye positions for each participant and do not take into account the time. Although participant gaze points plotted separately, it's hard to distinguish whether gaze points are looking at the same point at the time. The figure gets more complicated when we plot it as time series. It's not obvious to see whether multiple gaze points match. However, when we correlate these series with a tolerance value, we can distinguish the pattern that we provided to the participants. The three-dimensional plot is not very clear as well, but if we have chance to interact with the data, it's possible to see the pattern. Here is a short demo of collected data and the video. In the second part of my presentation, I want to talk about a better method that we use to quantify the recurrence of multiple users' gaze points. Let's assume that we have two participants looking at a two-dimensional space. Our temporal domain is 10 units of time, and we define the recurrence when the distance between participants' gaze points are at most 1 unit away. The XY plot on the left-hand side demonstrates a random gaze coordinates for two participants. First participant is illustrated by red and the gaze point started at number 1 in red on the top left of the figure. The other participants' gaze started from the opposite direction and illustrated by colored blue. Each point has 10 time steps, and the figure on the right hand side illustrates the recurrence plot of these events. Let's look closer to that figure on the next slide. Each axis represents a single participant. In this case, x-axis represents participant 1 and y-axis represents participant 2. When participant's eye in the start position, which is number 1, the other participant's gaze is on the other side of the area. If we draw a circle with radius of 1 unit, this will never match to another gaze coordinate. Therefore, first column of the plot will not represent any recurrence. However, when we iterate to the next step in the temporal domain, first participant's gaze point matches with multiple positions of second participant. Then it became easier to distinguish the distance for recurrence when we use color to represent spatial relation. We can clearly see that first participant look at the position at first, and then 2 time steps later, the gaze point of participant 2 gets closer by 2 units, stayed within the distance of 1 for another time step, and exits the recurrence distance. We calculated the rate of recurrence at 10% for this graph. Let's get back to our experiment that we presented in the first part of the presentation and use that data to plot the following figures. Each figure represents pairwise recurrence quantification. The figure on the left compares participant A and B and reports that recurrence is around 13%. The figure on the middle compares A and C, and the result is 14.8%. And the last one is between B and C and represents the highest pairwise recurrence of 15.5%. So what is next? First, we plan to refine and enhance our visual analysis techniques. Second, we want to discover data analysis part in detail. And finally, we want to design and conduct formal experiments to collect real data for the next phase of our research. Thank you.